Thank you for having me here. It's an opportunity for me to talk to American students. I always do Israeli students. It's new to me, so it's in a way another new thing here. So I would like to talk with you a little bit about the challenges that we face in product development and how, in my opinion, we should look at it not only from the research perspective, but also how we deal with the market, how we get to the market, what is important during development in order to be successful. So first of all, stem cell therapies are researched from two angles. One is the research side of it. Where do they come from? What can they do? For example, embryonic stem cells came from embryos that were not implanted back through the in vitro fertilization processes, and their mothers donated them under official informed consent. It is controversial to some religious people and to some not. But in any case, from biological point of view, we have two different specificities to these cells. One is that they can become whatever we want, because this is how we all began, from fertilized egg. So it means that they have the ability to become each and every one of the cells in our body and create tissues at the long run. It doesn't happen right now in the lab. Usually what we can do is differentiate those cells into neural-like cells, into muscle-like cells, and we hope that when we implant them back to the body, they will become the cells that we want them to become. I'm not working on embryonic stem cells, and when I say we, I mean all the scientific community that dealing with stem cells. But on the other hand, these same cells, since they are meant to create the whole body within 9 months, they proliferate, they multiply themselves very fast. And this is posing a risk when you inject them to a patient, because if they will do this over multiplication, it might pose a risk of cancer in the transplanted patient. So they are very promising, but still there are basic features that need to be solved in order to create a medication out of them. On the other hand, we have what we call adult stem cells. Adult stem cells, it's not if you are matured and did your test. It's if you were born. Already when you are born, your stem cells are adult because they are already organized and differentiated into functioning tissues. For example, your brain has neural cells which are fully matured and functioning. This is why we call them mature. So when we take those mature stem cells, obviously most of them are not as what we call plastic, cannot become everything that we want. And they cannot multiply themselves endlessly. So it means that we can use them for less things or more specific things, and they pose less risk from the point of view of whether they will create cancer or not. And I will come back to that in a minute when I will talk about what the regulator thinks, because this is the way the regulator, the FDA, who is responsible to our safety when we produce, when we give new products to the market, this is how they think. What is the risk that a new therapy will pose to the population, and what are the benefits? And we try to weigh them in a way and say, would we do it? And if we do it, what is it that we will ask from the companies to show us as supportive data that what we do is safe and of course efficient. So this is the area of product development. Not only researching the science of these cells, but how we use them, how we take them, and we bring them to market. And the market is only at its very beginning, and I just put here different type of companies that really do cell therapy. either, for example, Osiris, from donors, like one person, they take its bone marrow, and Pluristem takes it from placenta of an embryo after it was born, different sources of cells, but one donation, and then they multiply the cells in order to then put them in vials and ship them to the centers, to the clinical centers, and we call it in my language allogeneic, which means it's not of my own cells, it's from someone else, it's from a donation. So they are after somewhat of the model of pharmacy nowadays. You cook all the pills in one big place, and then you just spread them all around to all the pharmacies. So this is the model of what we call allogeneic cells, one donor multiplying the cells in a lab and then packaging them and shipping them to places where they are needed. And on the other end, we have companies like Astrom and like the company that I will represent today, Biogencell, and we deal with cells from the patient himself. So we actually take your own cells, we create your own medication to you in what we call personalized medicine. Most of these companies, like Astrom, take their cells from the bone, from the hip. And other companies are developing, like Baxter, for example, they have machinery for stem cells, and Satori have machinery, like they are not doing stem cells, they are doing the machinery that enables us to purify cells or select cells or take cells from the body, different sorts of what we call supportive instruments, and companies that have big manufacturing places where they can manufacture for themselves and for others. So there are different ways to look at the market, but all of them related to the stem cell market. So let's just try and understand. What I try to figure out here is, when you have mass treatment, most of us have, when you have personalized medicine, and how it goes with the severity of a certain disease. So most of us will take drugs. Like when we have headache, we take Advil, I do. And this is the same pill for everyone, and we all take it, nothing personal, and actually we are not very sick. And as it goes with very sick people, then they get to get personalized medicine. It's an operation, the patient is on the table, everyone is around him, 5 people or even more, super personalized medicine that we know today. And cell therapy right now is in this stage. Since it is relatively new area, we do not expose at this stage people who are not very sick to the therapy, because again, and I will repeat it in different angles, risk benefit, they can only benefit from it. There is a risk, but they will die anyway. So either we will benefit them, and by that we will learn about the cells and their activity to the long run, or we can't help them, but at least we did not risk any person who is not severely ill. But in the future, stem cell therapy will be everywhere, because stem cell therapy enables you to regenerate your function and your body, which makes you more energetic and makes your skin look younger. So, of course, you can imagine through cosmetics and everything else, we can use stem cells. So actually, it's providing patients with these unique needs. So what are stem cells? And I'm trying to say it, and of course, always it's so so complicated. They have actually two very basic features. First of all, they can renew. Renew meaning that the cells, when one of them go out because he is needed to regenerate a tissue, and it happens in our bodies all the time, he will first duplicate himself, leave his son, probably in the bone marrow. Most of the stem cells are in the bone marrow. He will leave its son in the bone marrow and he will go away and start to differentiate, to become an expert in what is needed. If the cell is needed in a muscle cell, he will become a muscle. If he is needed in a neural cell, he will become a neuron. And this actually happens along the way. I'm sure you have been told for so many times that the road is as important as achieving the mission. So this is really, in science, you can follow the cell and follow its maturation along the way, and you can see it happen. This is actually in the basic research, but we know that and we take advantage of the knowledge that we get from this science. So Professor Dov Tzipori, in his paper of 2005, tried to ask, what are stem cells? Okay, we know they have the self-renewal and we know they can differentiate and specialize along their movement in the body. But what are they? And he came to the conclusion that they can go back and forth and actually become expert or multiply themselves unless they really come into a tissue and then they fully differentiate and function, and then, like every other cell, with time they will die. But since this can happen in all the ways, like I can take a cell from a tissue and what I call under relatively good pressure, make him to go backwards and then specialized otherwise, which means this arrow, he came to the conclusion that stem cell is more of a status than a definition. Like you can change your status. And these cells has these features. They can home, they can penetrate through tissues from the blood where they come from, through tissues to the place they are needed. And we know how to trace this in culture. They have the plasticity to become something a little bit else, and we said that adult stem cells can do it to a limited amount, not to all the tissues. And then they can regenerate the tissue, but another thing they do is that by secreting molecules out of themselves, because this is how these cells work, they can also ask the blood to recruit some more cells to the area. And as long there is a damage in the tissue, stem cells will continue send these signals to recruit more and more cells from the bone. Now I like this picture because it usually shows very clearly that the bone is the safe of our body. It is strong, it is protective, but if you look inside, you will see that it is full of holes and many blood vessels come across it. So there is a very intensive crosstalk between any of my tissues and my bones. And the stem cells are laid here in these niches. We call it niche because it's like a very small room. And when we need them, they go out and then they differentiate to whatever is needed. Just one sample of taking these cells and treating people who suffered acute myocardial infarction, heart attack. And this German guy, Strauer, as I told you, it treated patients who were very severely ill, and because the outcome was very good, as I'm going to show you, we were able not only to see the improvement on the patients, but also to understand what happens to patients 5 years after treatment, one single treatment. And what we can see here is, first of all, mortality. How many people dies after heart attack. So he is a clinician, and he asked people who were under this situation whether or not they want to get their own bone marrow transplantation. As we said, autologous, my own. And 201 say, no, thank you, we're okay, we will go with the usual procedure. And 191 said, we do want to try. And see what happens 5 years later. This is the regular mortality rate, 4% a year, meaning that 32 out of these 200 died during these 5 years, as opposed to only 7 of 191 patients that chose to get the therapy. And this is where I usually laugh and say, okay, thanks to it, we can really know what happens on the long term. 5 years later, whether they have cancer or not, we can follow that. We cannot follow it in people who died. So first of all, it enabled us the opportunity to see and to follow them. And then we have scoring of how diseased you are. For example, they ask you how many stairs can you climb. Usually, these patients can almost not do it. How many pillows you need under your head in order to breathe at night when you go to sleep. All these questions that enable the physician to understand how severe the patient is. And we call it scoring because it's not a real measurement like we measure our BIPs. And all this scale is 0 to 4. All of you here are 0. And a very sick person would be 4. So we can see again the natural cause of the disease. The patients become more severely ill. Their scoring is raising. However, when you treat with stem cells, not only you did not stay on the same level where you have been, but you decline, you feel better. Declining means that you reduced your score by 1.2 points of scoring, and these people feel better. If we look at the last thing that he tested is the volume that the heart puts out in every beat. You know that we do it many times, actually 74, Mike, correct me if I'm...
Average is 72 beats per second, per minute.
72 times per minute, and in each beat, the heart sends blood to our body. So we have what we call ejection fraction, which means what is the fraction of the blood that is going out. And as you can see, and again, very typical to the disease, this is becoming less and less effective with the disease, meaning that the heart has hard time to send the blood out to the body. Whereas in these patients, it increased. So this is what stem cells can do. They can really help quality of life and functionality of the tissue. So what is the ecosystem here? And because, okay, this is nice, but we need to bring it to market. We need to understand what is the ecosystem, and the ecosystem would always be patient and physician, diagnosis, sampling, taking some samples, and then someone has to finance the treatment, insurance company or the patient himself, and this belongs to the model that each company will develop. And someone has to manufacture the cells, to create the cells. And then we have again patient and physician. There is a need to treat them. So the vision is always, I think, to all of us, it doesn't matter that I'm talking here, I think everyone would say the same. Affordable, highly standardized and accessible products. And the means are always the same. Try to become a factory. See how you go from a very specific process in the hospital, deliberately doing steps A, 1, C, and D to patient A, but A, 1, D, and F to another patient. This is not standardized, and this cannot be scaled up. What we need to think about is how you do it in a standardized way that can be scaled. And usually I'm trying to say what is it that we need, and actually it's the whole world. It's first of all market definition and looking at existing players and what they do. Product definition, what exactly will be the product that I want to develop, and development stages and the cost of the development stages. All of these should lay on regulatory demands and manufacturing process, meaning what are my limitations and what should I stand for when I do that, and manufacturing cost, reimbursement, how is this viable, and of course competitors. Every story has its own personal touch, mine too. The patients we are talking about, those who are heart patients or severely blood vessel malfunction patients, those are usually chronically ill patients. They are ill for many years, and they go to the clinicians, and every time and again they are being taking their blood samples and having them going through CTs and all these things for years. They are actually not only chronically ill patients, they are tortured on almost a daily basis. So what we wanted to do is, of course we want a safe and efficacious product, it's for sure, otherwise we won't be here. But we wanted to find the harmless way to acquire the cells, because we work, as I told you, on a personal basis based on the cells from the patient himself. And then we wanted it to be with long-lasting effects, so I will not need to treat them again and again and again, just one time. Of course accessible, and I don't want them to drive, as Mike said before with the women, I don't want them to drive to another country or to only one hospital. I want it to be everywhere. And all of this has to be capable of scalability and to be with cost effectiveness. So the product I'm going to tell you about today is such a product that is trying to answer all these questions or this wish list. And the reason I did it, as I told you, was a little bit personal. This is my late mother. My mother died 2 years ago, and she was diabetic, chronically ill, after heart bypass surgery and artery bypass surgery, without an ability to walk more than 5 steps. By the way, she, as I told you, we needed to be scalable and standardized. She insisted 2 months before she died, she was already on dialysis. She said, you're going to take my blood and you're going to show the world that you can do it from even a patient like me in my situation. So this is her, and we did take her blood, and we did make the same product as we did from my blood, from my mother's blood. We couldn't treat her, of course, because we weren't under clinical study at that time, 2 years ago. And this is my best friend, mother, who died actually a few months before my mother. They died almost together. They were very good friends. When she died, she did not have her 2 legs already for a few years. So again, these are the patients that we have in mind when we develop our product. 5. So it needs a teamwork to do that, and we need to understand what are the processes that we need to go through and the regulations and how the regulator will look at our production process and what he will demand from us. And this is what we do, because I promised I will show you what we do. So we are dealing with blood vessel malfunction, which means that the very tiny blood vessels are blocked, and therefore blood supply does not reach the organ. In this case, it's the leg. And since it doesn't reach the organ starts to deteriorate, they cannot walk, and then their leg becomes swollen, and they have cuts, and the cuts contaminated, and they have unhealed wounds and gangrene, and at the end of the road, we have to amputate them. We have to take their leg off. And what we came with the solution is to take just a very simple blood draw. As we sit here, I could ask from any one of you to give me a blood draw, and this is what I do to these people. We take 250 mLs of blood, and Mike will tell me the ounces.
8 ounce cup.
And
less than a can of soda.
And within less than a day, we create a population deliberately changed to become cells that can regenerate blood vessels, and we pack them in a syringe ready to use, ship them to the clinician, and he will inject it to the patient leg. This is how the artery look in the patient leg, and this is what our cells do. They create bypasses to overcome this blockage and resupply the tissue with blood. And this is how it looks in an animal study. What we did is we cut out part of the artery at the beginning of the leg, and if you look at the laser Doppler, this is before the damage. You can see blood supply to the leg, to the tail, and to the other leg, and immediately after we can see that the leg that was injured is black because it doesn't get any blood supply. When we look at it 15 and 21 days later, we can see the control group as opposed to animals that were treated with our cells. And as we said, what they do is regenerate new vasculature that overcome the problem and resupply the leg with blood. And if you can see the brown dots here, each of these dots is a new capillary, and this is how it happened. This is the control. So on the long run, we start with manually manufacturing of these cells, aiming to get very low amount of the market at this stage, and then go to fully automated production process. It's a coffee machine because we don't have the product yet, but we envision it. And we do envision it exactly as a coffee machine with the buttons where you pick your coffee, and I will pick heart disease, stroke, leg disease, and the machine will create the cells as I want them. And this is the benefits to us, to the health community, because using that will prevent amputations and therefore will reduce healthcare cost, and to the clinician, and of course, the main issue, to the patient. And with that, I would like to thank you for your attention.